Welcome back, everybody. And here is part 2 of our Chapter 11 Fundamentals of the Nervous System and Nervous Tissue lecture. Let's get started, picking up right where we left off. We had some terminology here at the end of our last lecture. Now let's talk about how the nervous system actually works. Okay, so we've talked about all the characters. Now let's talk about the plot. How does this actually work here? It starts out, and it is only possible because of what is called the resting membrane potential. Okay, so the resting membrane potential is basically where all of your neurons are when they are at rest, when they are not sending a signal, when they are waiting for their next job, right? And if you don't remember, a membrane potential specifically, is a difference between the inside and the outside of the membrane, specifically a difference in charge, okay? So there's a difference in positive and negative charge across the membrane from the inside of the cell to the outside of the cell, and it is accomplished by ions, right? So again, cations, positive ions, and anions, negative ions, those are what give the two sides of the membrane its charge. And at resting membrane potential, you have, this is very important, a higher positive charge outside of the cell and a higher negative charge inside of the cell, okay? We can then say that that cell membrane is polarized, right? Because something that is polar has two different ends, right? Positive end and a negative end. So if the membrane has a resting membrane potential, if that electric potential on either side of that plasma membrane is more positive on the outside and more negative on the inside at a specific level, then that cell is ready to fire, okay? So most of the neurons in your body, when they're not being used, sit at resting membrane potential, and that's when they are like at their greatest potential, right? They're at their most potential. And just to give you an idea, in case you're more chemistry or physics minded, what that actual potential looks like, we're talking about positive ions being more abundant in the extracellular fluid, or the ECF, and negative ions, or just fewer positive ions, being present in the intracellular fluid, or the ICF. In this image here, you can see the wall or the plasma membrane of a neuron. So imagine this being like the axon. If you snipped an axon and then looked down that tube, right? This is what you would be seeing. You'd be seeing more positive ions, namely sodium here, and fewer negative ions out here, and lower positive ion concentration inside of that axon, okay? So it just because potassium is positive, it'll make sense in a little bit, but there are also going to be other things in here like chloride, which are negative anions, negatively charged anions in here as well, giving the inside of the cell a negative charge. It is the sodium ions in the extracellular fluid that really give the outside of the membrane that positive charge. So you can see this is in millimoles, okay? So you have 50 to the minus 15 millimoles of sodium cations in the intracellular fluid. That is like 0.0000015 zeros 5 millimoles of sodium on the inside, so very, very small amount, and 145 millimoles of that same cation on the outside, right? A larger amount of potassium on the inside than on the outside, but again, that is balanced out by things like chloride, which is like here. These negative anions have a higher concentration inside of the cell than they do outside of the cell. This is nanomoles, in case you're curious about that. Let me just put in that little space. Okay. So generally speaking, you're going to have a negative charge on the inside and a positive charge on the outside when the membrane is at rest, when that cell is at rest. And we call that resting membrane potential, okay? So the way that resting membrane potential is maintained by these axons, and it's why the way that it is brought back to resting membrane potential after the axon has been sending its signals, is by sodium-potassium pumps, which we mentioned back when we talked about cells and how cells work back in part A. Sodium-potassium pumps specifically are one of those pumps that are able to send something out and something in at the same time, right? Using that secondary active transport. So basically what you have is a protein embedded in the membrane, right? That is it's a pump. So it's going to be changing shape to move a substance, a solute, from one side of the membrane to the other side of the membrane. And what a sodium-potassium pump does specifically is it pumps sodium out of the cell, so that is all on the outside, and it pumps potassium in, so that potassium is on the inside, along with these negative anions that are pumped in by other means, okay? So remember we talked about like gradients. So there's electrical gradients, right? Which we're not really creating here, but there's still chemical gradients that we're creating here. Less sodium on the inside than on the outside means that sodium wants to get in. So we can maintain a certain amount of positive charge on the inside, not enough to leave resting membrane potential, but enough for like homeostasis by using a different cation, potassium namely, while also maintaining the chemical gradient that keeps sodium wanting to get in, which is what's really important here, okay? And you'll see why in a second. So the way the sodium-potassium pumps work step by step, basically, is that the intracellular fluid sodium, the sodium in the intracellular fluid binds to the pump. The pump uses ATP, because this is active transport, uses ATP hydrolyzation. That chemical reaction changes the shape of the pump of this protein. That phosphate group that leaves as a result of that hydrolyzation is going to bind to the pump. That sodium, that pump changes shape, the sodium leaves, goes into the extracellular fluid where we want it. We're pumping it out because we're trying to get to resting membrane potential. That's this guy's job, right? That sodium is released to the extracellular fluid. Then the potassium that's in the extracellular fluid, these guys right here, are going to bind to the pump. That phosphate group is going to release from the pump, from that protein, and then that's going to cause the protein to change shape back to what it was before, which releases the potassium into the intracellular fluid. Okay, so long story short, sodium-potassium pumps pump sodium out of the cell whilst pumping potassium in, pumping sodium out and potassium in, using ATP because it's active transport. Okay, so resting membrane potential is going to result, again, in a polar membrane. It's going to result in a higher positive charge on the outside than on the inside of the cell, okay? So as you can see here, if we were to actually measure this, you could see that the cell has a -7 millivoltage, minus 70 millivolt voltage on the inside, whereas on the outside there is a 0 millivolt voltage. Okay, so the intracellular fluid is slightly more negative than the extracellular fluid, and -70 is that is accurate for a lot of your neurons. Okay, so the extracellular fluid has a slight excess of cations, right, and the intracellular fluid has a slight excess of anions. Okay. Okay, so the resting membrane potential is maintained by other gated channels as well. There are 3 different types of gated channels besides the sodium-potassium pump. You have your chemically gated channels, which are channels that are triggered by receiving a ligand or a chemical or a particular molecule, right? That's what causes it to change shape or to open. You can also have voltage gated ion channels, which are literally opened when they detect a change in voltage in the membrane which they are embedded in, okay? And finally, you have mechanically gated channels, which are so cute and funny, I feel like, because they have to open from like literal like physical touch. So it has to be an actual pressure applied to the membrane, a physical touch applied to the membrane for those little channels to get squeezed open. Kind of weird, right? So they open in response to pressure. So it's 3 different types of channels that are used to maintain the resting membrane potential of various nerve cells, various neurons across the nervous system. Okay, so when you're in resting membrane potential, you're just chilling out, right? When you get a signal from another neuron or from a sensory organ, then you need to wake up and do your job, right? What that job starts out as is, of course, taking that signal from your dendrites or from wherever the synapse is. It could be at the end of the dendrite, but it could also be actually on the body of you on the cell body. But regardless, wherever that synapse is, that signal is received at the end where the cell body is, right? That signal gets transported through the cell body to the axon hillock. So recall your neuron has like a little star shape and a long tail, right? This is the cell body. The signal comes in through this direction always. The signal going from basically here to here, where is the axon hillock, is what is called a graded potential. It's actually slightly different than the signal that is sent from that point on to the next cell. A graded potential, generally speaking, is more variable in strength, okay? It can only travel short distances, which is important. And as it travels, it loses strength as it travels through the cell, okay? So it kind of diminishes as it goes from the dendrites to the axon hillock. If it diminishes enough, it disappears completely before it gets to the axon hillock, and that's basically where it dies. And that signal does not get passed on. It the axon hillock or that neuron decided that cell or that signal was not important enough to pass on, okay? Because it didn't cause the axon hillock area to cross a threshold, causing it to send that signal on down the axon, okay? So the graded potential is the input signal from the dendrites. It usually starts the dendrites, but it could start like closer to the cell body, anywhere. They're just kind of they're touching each other like this, right? The axon terminals of one neuron and the dendrites of the other, they're kind of just mixing like this. So you have multiple synapses basically. Wherever that signal came from, whatever synapse, that signal needs to transfer through the cell body before reaching the axon hillock, okay? The gated channels that can cause a graded potential to start in the body of the neuron can be mechanical, chemical, or voltage gated, okay? The ions that are usually involved, sodium, potassium, but also calcium. And the signal can be depolarizing or hyperpolarizing. These are little details that'll make more sense when we get further along in this conversation. But most importantly, you are going to have a loss of strength as it travels through the cell. It may or may not reach the axon hillock with enough strength to cause the neuron to cause an action potential, which is what we call the signal from the axon hillock down the length of the axon all the way to the axon terminals to send that signal to the next cell, okay? Action potentials are very short, but they are very strong depolarization, okay? So basically remember at resting potential, the cell membrane is polarized, positive on one side and negative on the other. That it's depolarization that is the signal, okay? So the action potential causes a large depolarization that can travel a long distance. It can travel all the way down the axon to get to the next cell, okay? And it doesn't lose strength. It keeps getting replenished basically down the length of the axon, okay? So these guys it's a regenerating conduction signal based on the graded potential crossing a threshold of energy at the axon hillock. It's going to come from the trigger zone in the axon, which is usually in the axon hillock again. Okay. These signals come from, and the signal of a signal of an action potential is sent via voltage gated channels, specifically using sodium and potassium, as we've already discussed. We are using depolarization again, and the strength is basically all or none. So you either have an action potential or you don't. There are no weaker and stronger action potentials. They're all the same strength, though they can come at a greater or lesser frequency. Okay. Okay. A threshold stimulus is required to initiate an action potential. All right. So here is the presynaptic neuron down here. This is the presynaptic neuron up here. Pardon me. So it got a signal through its dendrites. That signal ended up reaching the threshold in the axon hillock of this presynaptic neuron. It decided to start an action potential down its axon, and now that signal is reaching this postsynaptic neuron, okay, which is this guy right here, okay? So here is that axon terminal from that presynaptic neuron right there, sending that stimulus to the postsynaptic neuron. Some of that current is going to leak out of the cell, hence it losing strength. This is a graded potential losing strength as it travels to the trigger zone. Enter the trigger zone, which is in the axon hillock again. Okay, so if that graded potential doesn't make it all the way into the trigger zone, or if it's not strong enough once it reaches the trigger zone, that's the end of that signal, and we refer to it as a sub-threshold graded potential. If it does make it to the trigger zone, if it is strong enough to make it to the trigger zone and cross the threshold at the trigger zone and then the axon hillock, then it is referred to as a supra-threshold graded potential, and it will stimulate an action potential to be started down the axon, okay? So once that graded potential passes through the cell body and reaches the trigger zone at enough strength to surpass the threshold, that is a supra-threshold graded potential, then the action potential will be activated basically at the trigger zone. And as we go down the axon, it will be continuously like rebuilt as we go down. So basically, if you're measuring the signal down the length of the axon, that signal will continue to be strengthened each time it passes a certain region of the axon. Okay, so as we go down, it continues to be like replenished. Okay, this is because those voltage gated channels are all the way on the plasma membrane, all the way down the axon. So those channels are going to continue to depolarize that cell as we go down. It'll feel that the depolarization from ahead of it or from behind it, and it will depolarize and send that signal along ahead of it. Okay, so unlike a graded action potential, it doesn't lose strength, basically. Long story short, okay? So you've got some, if you were to measure this signal as it passes one point in the axon, what you would see is this one bump right here. Okay, so this is basically just this is one of these. Okay, so just one of these, if we were to take a closer look at one of these guys, right, we would see the entire cycle of depolarization. Basically, we'd see we'd see the main steps of that signal, and the steps are depolarization, which happens starting at resting membrane potential. Remember, we are starting with a polar membrane, positive ions on the outside, negative on the inside. You receive that depolarizing stimulus, which could be, if we were up here, that would be like that graded potential crossing the threshold here at the trigger zone. But right here, it really would be those voltage gated channels in the membrane right there at that particular node of Ranvier, if you recall, in between the two Schwann cells, remember? That depolarizing stimulus is coming from the voltage gated channels from ahead of it, okay, or from nearer the cell body. So once that threshold is reached, those voltage gated sodium and potassium channels open, and sodium basically floods into the cell, right? Because sodium has that chemical gradient that it wants to go down. So it's going to flood as soon as those gates open. That sodium is going to rush right in because it wants to go down its gradient, right? Its concentration gradient. And that rapid sodium entry is going to depolarize the cell. So basically, to depolarize is to go from being positive on one side and negative on the other to being roughly the same on both. So once it's depolarized, you have the same charge on both sides of the membrane, right? Then the sodium channels are going to start to close once we reach about here, right? So you've got resting potential, sodium channels open, sodium rushes in the cell. You have a great deal of membrane of millivoltage, right? So the voltage goes up. You reach a maximum at this particular point as it's passing by, as the signal is passing by, and then the sodium channels start to close, and those potassium channels are open fully. So the potassium channels are opening more slowly, basically. The sodium gates open real fast, sodium rushes in, the potassium channels kind of take their time, and then they kind of stay open for a little bit longer after the sodium channels close, and sodium starts to get pumped back out of the cell, okay? So potassium kind of follows along at a lag. Repolarization is kind of from this point forward, okay? So potassium starts to be moved out of the cell along with the sodium. The potassium channels stay open to allow potassium to kind of leak out against its electrical gradient as the cell repolarizes. We're trying to get back to positive on the outside and negative on the inside. So positive stuff is getting shooed away. Potassium is getting pumped out. Those potassium channels close slowly, and then the sodium potassium pumps get to work and start pumping that sodium out more quickly and forcefully to get back to resting membrane potential, back to polarity. We are repolarizing, okay? So the depolarization is the signal, and then we repolarize to get back to resting membrane potential so that we can get another signal. So if you were to look at the axon in a cross section right here and compare it to what the signal looks like on a graph, you would kind of see how the membrane potential is changing in a little section moving down the axon, okay? So that membrane potential is shifting all the way down the axon, and once it passes, the previous section recovers back to resting membrane potential, okay? Step by step now. So you've got these sodium potassium channels, you've got sodium channels and potassium channels. These are more like leakage channels that are not active transport. There's more passive transport, right? So all you really need once you reach threshold at the trigger zone is for that voltage to reach these voltage gated channels, and they just swing open for sodium. And since sodium has been corralled outside of the cell but has a low concentration inside, it rushes in down its concentration gradient into the inside of the cell. That is depolarization, okay? So the first thing that happens is the gate opens, boom. Now sodium, which once could not get in, now it can get in. The gate opens, sodium rushes through, okay? Sodium is rushing through. Sodium is rushing through in this part of the signal, okay? Once the signal has passed, this gate once the signal has passed, this gate, which is it's like a slower secondary, what's called an inactivation gate, will close. meaning that sodium can no longer go in. We're basically like holding space here for repolarization to eventually happen. This takes a little bit longer. That gate will eventually close. This one will open back up. Sodium still can't go in, but now potassium is still leaking out through other gated channels, okay? So that's just a step by step. You don't need to know all of those steps. I'm just trying to help you to understand. You need to know the basic steps. Basically, sodium rushes in to depolarize, and then sodium gets pumped out to repolarize, okay? All right. So here's another depiction of this, basically. So the action potential is traveling through time right here, the rising phase, basically going like this, right? So you start out with the depolarization at the trigger zone, triggering the sodium gates to open quickly, rapidly. Sodium floods in. You've got more depolarization as the next voltage gated channels feel that change in voltage, and then they open, and then sodium floods in, et cetera, et cetera. Once that signal has passed to stop the cycle, the inactivation gate of these sodium channels closes to no longer allow sodium to rush in. The potassium is allowed to leak out. Still, after that inactivation gate closes, the potassium still leaks out against down its concentration gradient now, basically just continuing the repolarization back to positive on the outside and negative on the inside until we get to repolarization. And those sodium potassium pumps get to work to really get us back to that minus 70 millivolts on the inside compared to 0 millivolts on the outside. Okay. Another picture showing this time the refractory period. So because of the time that it takes for these activation and inactivation gates to close and for the molecules, these actual ions, to travel through the gate, there is a period of time when that section of the axon can't be activated again. So it needs a little bit of time to reset, basically, okay? So that reset is called a refractory period. The absolute refractory period is in pink here, and that is when basically the neuron is busy, okay? It's busy already handling a signal. So in absolute refractory period, we cannot receive another signal because we're working on one right now. The gates are as open as they're gonna get, okay? So that's when the neuron cannot be excited again under any circumstances. The relative refractory period, which is in purple here, is after enough time has passed after the signal so that the neuron may be excited again, but only if the stimulus is stronger than the one that it just had. So we're not necessarily all the way back to resting potential yet when we get here. But with a strong enough signal, we could send that up again. But it has to be a stronger signal than the first one, okay? So the absolute refractory period, we absolutely cannot do another, we absolutely cannot have another signal. We absolutely cannot re-excite that neuron or that portion of the axon. And a relative refractory period is, we probably can't, but if it was strong enough, we could. Okay, so the way that the signal is propagated is most straightforward in non-myelinated neurons. So check it out. When this signal is a graded potential, it's traveling from the dendrite to the axon, right? But when it's traveling down the axon, we are going to see, oh, sorry, this is the graded potential. Sorry. My bad. Hang on. I want to make sure that I get this right. This is technically a graded potential because it is coming from the dendrite and having to pass through the cell body. As long as it's having to pass through the cell body, then that signal can be lost, okay? Fine, whatever. So the stimulus starts out strong when it enters the dendrite, but it loses power because it's leaking out across the membrane before it gets to the trigger zone, right? If there's still enough left when it gets to the trigger zone, then that neuron will start an action potential down the axon. If not, then it is going to be sub-threshold graded potential, and we will not get an action potential, and that signal will die there at the axon hillock, okay? Non-myelinated action potentials down the axon, okay? These do not lose strength because you have voltage-gated channels throughout the membrane all the way down the axon. So as soon as the stimulus causes this gate to open and sodium to flood in, that voltage change will then trigger this one to open and sodium to flood in. Then that voltage change will trigger this one to open and sodium to flood in, et cetera, causing the signal to maintain its strength all the way down the length of the axon and then be passed on to the next cell. This is when we are non-myelinated, right? So we don't see any Schwann cells wrapped around this axon. This is a bare, naked axon, right? Compare this axon to this axon where you've got these little bumps on it, right? Those are those Schwann cells from our previous lecture, remember? They wrap around the axon in the peripheral nervous system to form a myelin sheath, and that is basically an insulation around the wire that allows the signal to transfer more quickly, faster down that axon. So this is in the peripheral nervous system because these signals need to go a long way, right? These nerves of your peripheral nervous system need to travel from the central nervous system, from your spine, all the way out to your extremities or your organs, which is a relatively long distance. So we myelinate, we insulate those axons to let the signal go faster and to not be lost. Okay. So where there is a myelin sheath wrapping the axon, it's not exposed to the extracellular fluid. And that's where the sodium ions are, right? So if you've got your little sodium ions floating around in the extracellular fluid, they can't get through the myelin sheath, right? So you don't have any voltage-gated channels there. Instead, you just have voltage-gated channels in the gap, also known as the nodes of Ranvier. Nodes of Ranvier, because some French guy discovered them, right? So the signal is stimulated in one of these nodes, right, way up by the axon hillock. The signal gets passed underneath the myelin sheath and then gets replenished at the next node, okay? And the myelin sheath helps to insulate the wire so that the signal can go really, really fast. We basically call this saltatory conduction as opposed to continuous conduction. So saltatory conduction kind of goes in a leapfrog effect underneath each myelin sheath and gets replenished at each node in between the myelin sheaths, okay? Okay. Saltatory. Okay, so one more time. Continuous conduction travels down the axon and is replenished continually because there's nothing to, or because there's voltage-gated sodium channels all the way down the length of the axon, right? So that signal travels all the way down the length of the axon. The refractory period that occurs after that signal has passed is what guarantees that that signal can't go backwards, right? The refractory period is a simple fact of physics, but it is also extremely important because it allows for that signal to not go the wrong way. As long as you have a refractory period following the signal, that those voltage-gated channels can't open in the wrong direction. That would be terrible, right? So the refractory period keeps that signal going in one direction, which is really important. Okay. Versus saltatory conduction, like I said, it's the same basic thing, except you don't have these voltage gated channels underneath the Schwann cells, underneath the myelin sheaths of the Schwann cells. Instead, you have them just at the nodes of Ranvier. So the depolarization gets replenished at each node, and then the signal travels quickly all the way underneath the myelin sheath to the next node where it gets replenished. Okay, so the myelin sheath prevents loss of voltage by preventing current leakage. The sodium gated channels are concentrated in the nodes of Ranvier. MS and ALS are diseases that degenerate the myelin sheath. So like you could think about multiple sclerosis, if you know anybody that struggles with that, or ALS, they have a hard time controlling their movements, right? Because that signal keeps getting lost out of the axons of their peripheral nervous system neurons because they don't have the myelin sheath. So in these places where there's no myelin sheath, but there's also no voltage gated channels, you can lose voltage out of the axon as the signal goes by, meaning that the signal is weaker by the time it gets to the effector, whatever it is. Okay, so once that signal reaches the end of the axon and gets into the axon terminal, let's talk about how it gets passed on to the next cell. That gap in between, or that place of transfer, that junction, is called the synapse, okay? And it goes from the axon bulb, which is one of those axon terminals, right? It passes between that bulb through that part of the membrane of that neuron, of the presynaptic neuron, right? Which is also known as the presynaptic membrane, right? So it's just the membrane of the presynaptic neuron. It passes through, the signal passes through the synaptic cleft, which is the literal physical space in between the presynaptic neuron and the postsynaptic neuron. So the signal has to jump the gap and reach that postsynaptic membrane of the next neuron or the next cell, whatever it is, right? We talked about the neuromuscular junction, for instance. The presynaptic membrane would be that of a motor neuron, and the postsynaptic membrane would be that sarcolemma, remember, the plasma membrane of the muscle cell. Okay. Here's an actual picture of actual synapses talking to an actual cell body of a postsynaptic neuron. So this is a synapse, this is a synapse, this is a synapse. You see multiple synapses there, okay? Same words, different picture. Okay. Here's a close-up cartoony illustration of how that signal gets passed, okay? So the signal itself is actually what are called neurotransmitters, okay? So these little purple guys here, those are actual little molecules that get released from the presynaptic membrane and find their way to receptors on the postsynaptic membrane by crossing the synaptic cleft. Right? How this works is that the nerve impulse that was traveling down the axon, that action potential, right, reaches that axon terminal or the axon bulb or the, what was the other one? The terminal. It is going to cause calcium channels to open around on the surface of the bulb, okay? So that could be like around this part up here, over here, all the way around the bulb. You have calcium channels that are going to open, and calcium is going to diffuse into that axon bulb, into the terminal bulb, okay? Calcium diffuses in. then that is what causes these vesicles that are here waiting full of neurotransmitter at the synaptic, at the axon terminal, right there next to the synaptic cleft. These guys now, once the calcium comes in, will move to the presynaptic membrane, fuse with it, and release their neurotransmitter into the synaptic cleft, okay? Once these receptors on the postsynaptic cleft receive the neurotransmitter, which they are designed to receive, once they get that, they are going to open because they are chemical gated channels. Okay, so these guys are chemical gated channels in the postsynaptic membrane that open in response to receiving this neurotransmitter. The ligand for them that triggers them to change shape is the neurotransmitter. Okay, so sodium ions are going to diffuse in, starting a graded potential, right? Which needs to be strong enough to reach to the trigger zone in the axon hillock in order for an action potential to be generated in this neuron and passed on to the next neuron or cell, whatever it is. Okay, so those calcium gated channels they can be up here too. Okay, so they're going to be all over the bulb potentially, but once that calcium comes in, that's what signals these vesicles to fuse with the presynaptic membrane, allowing the neurotransmitter to go out into the synaptic cleft and bind to the receptors in the postsynaptic cell, okay, wherever that might be. This is going to lay that out again, basically what I just said. And then once that neurotransmitter has been sent, and once all of the voltage gated channels and we're in refractory period, et cetera, for this axon is all done, once the signal is passed through and we're done, basically, the neurotransmitter can be taken back up into the vesicles, or they will be taken up by glial cells, or they can go into adjacent blood vessels. So the neurotransmitter will be basically reabsorbed by multiple parties in the situation. It can be reabsorbed back into the presynaptic axon terminal. It can be reabsorbed by like astrocytes, which are in charge of maintaining this extracellular environment out here, right? So that's going to be part of their job. They can take up this extra neurotransmitter, or the neurotransmitter can make its way into the bloodstream, right? Diffuses out and away, because this is just a gap here at the synaptic cleft, right? Okay, and then once all of that neurotransmitter is absorbed or resorbed, then we're ready potentially for the next signal and the next release to continue that signal, right? Or to send a new signal. Okay, so these synapses can be chemical, which we just described, but they can also be electrical, which is kind of interesting. So electrical synapses, they're very rare. They're relatively rare. They're not terribly well understood at this point, but basically, instead of there being a synaptic cleft or a gap in between the presynaptic and postsynaptic neurons, you have the two bound together by gap junctions. So if you don't remember gap junctions, those are the ones where you have one cell wall, the other cell wall, and you basically make a tube in between them so that stuff can pass between cell 1 and cell 2. Okay. So instead of having a synaptic cleft, you actually have like passageways from one cell, from the presynaptic cell to the postsynaptic cell. So these signals can go very, very fast, which is important for like the central nervous system. So like this is used a lot for like actual thinking stuff, like memory and stuff. Your cardiac muscle uses this because the signal is needs to be very, very fast and very strong, and in case of cardiac muscle in particular, very consistent, right? Then chemical synapses are far more common, okay? Neurocrine molecules, basically, when we're talking about a chemical synapse, technically the neurotransmitter in that case is a chemical, which we call neurocrine, a neurocrine molecule. Okay. Here is a hideous chart of all of the different possible neurotransmitters, not all of them, but some of the more well-known and basic types of neurotransmitters. You do not need to memorize this. I wanted to put this here because it may be of interest to those of you that are interested in the nervous system. And I also wanted to give an example of how medications affect your body by affecting neurotransmitters and by affecting receptors, affecting the receptors here. So basically, an example that I was thinking about earlier is the neurotransmitter serotonin, right? Which is like the happy chemical, right? Think about antidepressants. There is a entire class of antidepressants called SRIs. That stands for serotonin reuptake inhibitors. So if you're looking at a synapse in a person's brain whose neurotransmitter is serotonin, that serotonin, that happy chemical, will be released and go on to the postsynaptic neuron, which is part of the chain of events that leads to a feeling of well-being and happiness, right? In people who are depressed, the idea is that there is not a lot of this neurotransmitter to go around, right? They have less serotonin. These synapses and the neurotransmitter that is used in their synapses is chemically, physically different. So the serotonin that they release as a neurotransmitter is either less, or it gets taken back up too quickly, or it doesn't bind to these receptors as well. Maybe they even have fewer receptors here. As maybe that's like that. Maybe that's the defect in the brain. It's really not very well known. But if they have fewer receptors here, then releasing the neurotransmitter serotonin will not be able to bind as well here. And then you're going to end up potentially with depression. What an SRI does as a serotonin reuptake inhibitor is it inhibits the reuptake of serotonin. So basically it causes serotonin to hang out in the synaptic cleft longer, which means that it has a better chance of binding to the postsynaptic cell and therefore have a better chance of behaving normally, if you will, in that person's brain. So as an example, something like an SRI, a pill that you can take every day, what it's actually doing is it's affecting the way that your synapses work. It's affecting the way that your synapses are able to interact with the neurotransmitters of your body. Medications can affect these receptors. They can affect the reuptake from any direction of a neurotransmitter. They can simulate a neurotransmitter and basically make more of it. They can cause the neuron to produce more of the neurotransmitter. They can cause a neuron to have fewer receptors for a neurotransmitter. A lot of medications work on this system, which is amazing because it's one of the systems that we know the least about, and it's kind of awesome that we have figured out it as much as we have about it, and enough to make medicines that can help people in this way. So it's really very cool. I'm still thinking about what to do about your guys' first discussion assignment. Obviously, I have to figure it out before I make it due, but one idea that I had is to have you guys choose a neurotransmitter, or choose a pharmaceutical drug or any drug and describe how it affects neurotransmitters, and what neurotransmitters and what kinds of cells and receptors does it affect. Because it's so fascinating, and I think it would be a really good exercise to get an understanding of how drugs work on the brain, right? All right, you guys, that's it for today. Thank you for joining me for chapter 11, the fundamentals of the nervous system and nervous tissue. And I will see you guys on Thursday for our first official lab. We're going to be looking at slides under a microscope, and I think dissecting a piece of brain. Not sure what type of brain yet. I'm trying to find out for you so I can give you more details on that. But if you want to prepare for Thursday's lab, I highly recommend that you go through the structures that we talked about in this PowerPoint, namely the important glial cells and the structures, the basic structures of neurons and the different structural types of neurons, where they can be found. This is very important, right? So types of neurons, structural classes of neurons, where they can be found, and types of neuroglial cells and where they can be found, central nervous, peripheral nervous system. That's going to be what you want to really focus on Thursday. Okay, so don't forget to crack your book open, take a look at this chapter, take lots of notes, especially again on these types of neurons and the neuroglial cells involved in both the central nervous system and the peripheral nervous system, and see if you see how much of that you can identify under the slides on Thursday. And next week, we'll talk about the central nervous system specifically, and then we'll do some more dissections to explore that some more, and histology slides and stuff like that. Be aware that your to-do list for this week and next, actually the modules themselves, do have details about your lab. This is the stuff that I'm asking you to take a look at the very least before we get to our actual labs on Thursday, which are described very generally here and here, these assignments. And again, your attendance and participation are what I base your grade on for the lab activities. So show up, be on time before I lock the door. Make sure you have your symptom checker app and your green check mark out so that I can look at it before you come in. Make sure you have a mask on, remember to social distance, and we will start our first lab. I don't know about you, but I am super excited for that. And so with that, looking forward to seeing you on Thursday. Have a good night.